Now that we've picked our colors and probably done so in way more ways than we actually needed to, we are ready to move into making the heat map. We have colors and we have to make sure we have our data, right? So that's the next bit of this, is to remind ourselves what the data is that we're coming into this clustering and heat map generation with. And so I have a code chunk here, but really this code is all stuff you should have already run in the step 5 script, and you should have these objects, results, diff genes, in your R environment. If you don't, you can just rerun this, but if you saved your R project, they should be here. It also just gives us an opportunity to kind of refresh our memory on exactly how this diff genes object was generated. So recall, we ran a decide test function from Lima that takes our Bayesian stats and does some correcting for multiple testing using Benjamini-Hochberg, and then applies some cutoffs. And we control those cutoffs. We picked a p-value, adjusted p-value cutoff of 0.01 and a log fold change of 2. So that's a fold change of 4. So pretty strict cutoffs. That then gave us this very nice way of kind of conceptually understanding how these cutoffs are used to select or test for genes that meet those cutoffs, because the result of decide tests is each row of our data is given a 0 or a 1. 0 means it failed that test. 1 means it passed, or it was differentially expressed. And if it's a positive 1, it was induced or upregulated in the comparison that we tested for. And if it is a negative 1, it was downregulated or reduced in expression in the comparison that we tested for. So we then used that decide test result to as a subsetting operation to pull out any gene in this single pairwise comparison, this single column, that did not equal 0. And we talked about how you could string together multiple of these sort of Boolean operators, effectively pulling out any portion of a sort of Venn diagram representation of your differentially expressed genes, or all differentially expressed genes, which is what we did. And when we finally produced that object, it had 2,314 rows, or differentially expressed genes, and our 10 samples. All right, so that's a data matrix, and that's important because the stuff we're going to do next really relies on having a data matrix, not a data frame. It needs to be numeric. All right, so we have our colors, we have our data, we are ready to start clustering. And we're actually going to cluster using a method we've already used in class. If you recall, when we were talking about exploring our data and doing multivariate analysis using things like principal component analysis, we actually started off that script using hierarchical clustering to just quickly understand relationships between our samples by producing and looking at a dendrogram tree that grouped our samples based on how similar or dissimilar they were. That same function, hclust, is what we'll be doing here. In fact, we will redo that clustering of our samples exactly, really, as we did it before, to understand sample relationships again, and to apply those sample relationships to our heat map plot that we'll produce. But really critical to making a heat map is actually clustering the rows. And so that's not something we've done yet, and we're going to do that now. So we're going to do that using the same function, hclust, and it's going to operate on a distance matrix, just as we do with our samples. That distance matrix is going to be created from correlations of our genes. And the correlations are going to be calculated using a Pearson correlation. And we'll talk about why and how we choose Pearson versus Spearman, and when one might not be appropriate in just a little bit. But I think it's important to understand that we're applying a correlation here. We're actually going to transpose our data. It's a small detail, but it's something we do when we're clustering the rows, because the core function is only going to work on sort of columns. And so if we want to cluster our genes, we have to sort of transpose them to be columns. So once we've done that, we've calculated the correlation, it's going to be a value that ranges from -1 to 1, -1 being, two genes are perfectly inversely correlated, and a +1 being perfectly correlated, and then all kinds of values in between. So we're going to take 1 minus those values to give us data that's on a scale of 0 to 2. That can then be used to calculate a distance matrix and then that distance matrix can be clustered using hclust. All right, and we have an agglomeration method here too, complete. And we talked about some of these agglomeration methods and hclust again earlier. Okay, hierarchical clustering, just to remind you, is an unsupervised clustering approach, similar to or related, at least in that respect, to other unsupervised clustering approaches like k-means and self-organizing maps. We'll talk a bit more about other clustering approaches a little bit later. Okay, once I run this line, I get a list object, cluster rows, a list of 7 elements that contains the results of that clustering operation. And I can then redo that clustering of samples as well. The only difference between this line and this line is that I'm not transposing because my samples are already columns, and that's what core is going to operate on, the correlation function. And I'm going to use a Spearman correlation instead of a Pearson correlation. And we're going to talk about why that is in a minute. The result I produce is another hierarchical clustering output, clust columns, still a list of 7. And it's just important to emphasize that we don't need to do this to make a heat map. In fact, some people prefer not to cluster their samples or apply a dendrogram tree to the top of their heat map because they want to force the order of the samples to be in the order that they're in the data itself. But there are other times where it makes sense to cluster columns because we want to show ourselves or the reader that the differentially expressed genes that we pulled out show some interesting relationships between our samples or highlight interesting biological relationships between the samples. So there are times where this clustering result for columns is useful and times when it's not, but it can always be generated in your R script and either used or not when you make the heat map. All right, now that we have this clustering of our genes and clustering of our samples, we're ready to assign modules, to produce modules from that clustering result. The clustering is done, but now we actually have to draw lines about which genes really belong in that module versus another one. And so to do that, we're going to use a cut tree function. And this is going to sound a little bit abstract when I'm talking through this, but it will make more sense once we see the heat map, and we'll put this into a bit more context. But cut tree is going to operate on the tree, the clustering tree that's produced from hclust of the genes, because we're doing it on clust rows. And it's going to cut that tree in a way that gives us 2 clusters. So first thing to realize here is we're setting that k equals to 2. Why? Well, we only have 2 phenotypes. You're either healthy or you have this disease. That's the data set we're working with in class. So by definition, we really only expect 2 clusters, genes that go up and genes that go down. Genes that don't go up or down presumably never made it through this differential gene expression analysis and should not be here. So we really are expecting just 2 clusters. What if, and chances are, you may be dealing at some point with a data set that has more than just one comparison, more than 2 possible modules. How do you decide on this k? Well, the answer that's probably not that satisfactory is there's no rule to determine what k should be, at least in this case. You are best to kind of iterate through and see what kind of cutting of your tree really makes the most sense. You can overcut and end up with many tiny clusters that really don't look that different, and you can undercut, which results in larger clusters where it's clear there's patterns of genes within those clusters that are behaving differently. Okay, we've cut to generate 2 groups of genes, 2 modules. And so what's produced from that is something called clust assign. That's what I'm calling it. And it's really a named integer vector here where each gene has its cluster assignment, module assignment, either module 1 or module 2. All right. Now that I have this module assignment, I can now use that to pick some colors that will be kind of mapped to these modules. And that's something aesthetic, but it's a useful aesthetic that will apply to our heat maps. So the way that works is we will run unique on the module assign. That'll look through this integer vector and see how many unique values there are. And of course, if we run this, there are only two unique values, the number 1 and the number 2, module 1, module 2. If I then take the length of that unique module assign, of course the length is going to be 2. Recall when we were talking about colors earlier that we often need to provide our palettes with an n equals something to pick a number of colors from that palette. So now this becomes really useful to say, no matter how many K clusters or modules we set up here, no matter whether you use cut tree or there are other functions to pick modules, no matter what you do, you can now automatically assign colors to it. So we will take the number we just generated and we'll pick that many colors from, in this case, the rainbow palette. Right? So when we run this, we now end up with something called module color, which is just a simple character vector of length 2, where each element is 1 hex code. We can now convert our module assign, which recall is this named integer vector, into a vector of our colors. All right. So when we run this, we get now module color has been overwritten, and now it's this. So it's a vector of length 2,314, the number of differentially expressed genes we have, where each of these is now sort of mapped to its appropriate color. All right. Again, this is an aesthetic thing that we'll appreciate, again, a lot of what we've done here once we make the heat map and can look at it and talk about these things in a bit more context, but they are useful things to do. Now that we have our colors, our assignments, our clusters, or clustering to produce the modules, and assignments of those modules to colors, we're ready to actually make a static heat map. And as I mentioned earlier, we do that using the heatmap.to function from the gplots package. I just find that a really convenient way to make a heat map. And it needs a few things specified here. So I want to talk through those and we'll make the heat map. The first, obviously, is it needs your data matrix, numeric data that it can actually make a heat map from. You don't have to give it clustering results to make a heat map, but we, of course, want to cluster the rows, and we're going to do that row v argument here as a dendrogram using the clustrows object we generated. Same thing with our columns, as dendrogram using clust columns. So it's going to apply a dendrogram to the side of our heat map and to the top. We then want to apply these module colors we just generated to the side of the rows. And then we now get to talk about what colors we actually want the heat map itself to be. And so we're just going to pick the heat map colors that we picked up above. This is one of the ones we made earlier. Now, important point here is that we're going to scale the data in the heat map, and I will talk in more detail about what that is in just a minute. We don't want to label the rows because putting 2,314 gene symbols on the rows of this will not make it legible. I turn off some annoying sort of annotation stuff that are default parameters in heatmap.to that I don't like, and then I set some row and column characteristics, font sizes, etc. here, and margins for the plot. All right, so let's just run this now. Enough talking. Let's see what this heat map looks like, and then we'll kind of review some of these things that we did. So the first thing to point out is this heat map looks really weird. It's very sort of, there's not a lot of blue here, right? And that's because I have scale set to none. It is plotting exactly the data that I passed to it. Log2 expression data for all 2,314 genes. It's applied a clustering tree on the left-hand side. That's my clust rows. It's applied a tree on the top of the heat map. That's my clust columns. And it's applied colors to those modules on the left-hand side. In case it wasn't visually apparent, that sidebar is here now. That's the row side colors, the module colors we generated. But again, I'm not really appreciating the changes in gene expression. Everything looks kind of red here. We're going to go into this in a bit more detail, but to show you what we really want to do here, we'd like to change that scale to be equal to row. And I'm going to recreate this heat map now, and now we're going to see something very different. Now, exact same data, we now have a heat map that looks very apparent, really striking, two totally different clusters, a set of genes that go up with expression, and a set of genes that go down in comparing controls to patients with the disease. Okay? All right. We'll again get back to this idea of scaling in just a second, but you can obviously swap out these colors. If you remember up above, we set up some color ramps, blue, white, red. We also did red, blue here. We have the GOT, Game of Thrones palette. Why not try that? And so we can add these colors into our heat map as we please, right? So let's try Game of Thrones. This is the Aria palette. Yeah, it's not great for a heat map as much as I liked Game of Thrones. And so you could kind of iterate through and try different colors. All right. Here is one of the other color patterns we picked. I think it's well, we'll see here in just a second. Okay, that's a red blue. You get the idea. Okay. So now that you've seen how to make a heat map, let's unpack some of these things about scaling the rows, about what the colors actually represent, and that'll be a good way to put all of this into context. But now you understand the clustering on the left-hand side, the clustering at the top, and when we ran that cut tree here, it was really just looking for a position on this dendrogram tree on the left-hand side where a vertical cut could be made to leave two branches. So obviously that would be cutting around here. You'd leave this whole branch, the orange cluster, and if you cut in this area right through here would be this other top cluster. If I change that to 3, it will cut the tree probably right here in a place to generate 3 distinct clusters. And if I choose 4, it'll move over to the right. So it keeps sliding to the right in a way to cut the tree that generates the number of clusters modules you indicated. And you can see if I chose 4, it would cut in a way that would leave this as its own sort of skinny separate cluster. And that's what I mean by kind of really thinking about how changing this number, whether it's really giving you something that's meaningful. Okay, so heading back to the slides, we've discussed our heat map a little bit, or made our heat map. Let's talk about a few things. The first is it actually matters what colors you choose for the heat map, as much as I joked around about the different color palettes. If you make red-green heat maps, just know that this is what your heat map will look like to people who have red-green color blindness. This is exactly the data we're using today. I just changed the color palette to red-green. So I tend to use red and blue colors, or blue and orange, whatever you prefer, because those will still be discernible modules, clusters, patterns to folks with red-green color blindness. So stay away from green-red. Have I ever used green-red heat maps in my papers? Absolutely. And I'm just trying to help you not make the same mistakes that I've made. All right, now let's talk about this difference between clustering our columns by Pearson versus Spearman correlation. If you recall, in that clust columns function, we used a Spearman correlation, but to cluster the rows, we used Pearson. So if we think about the sort of toy data set we were using earlier, I think we can highlight why that choice of Spearman for samples is important by adding an additional gene to this data set that is some funky gene called gene E. And gene E follows a circadian pattern as well. It's low in the day in the first day-night cycle. It comes up at night. It goes back down in the day. And then it goes way up in this second day-night cycle, but still follows a circadian rhythm where it's lower in the day and higher at night. Okay, so if we add that gene then to our map, this is what it would look like. And the question is then, what would this new heat map look like? And the answer is, well, it would really depend on whether which clustering method, which correlation method you use to generate your clustering. So if we use Spearman, even with this new funky gene introduced, our day samples cluster together and our night samples cluster together. And we can see this gene E clusters, because again, when we use cluster by rows, we use Pearson. We can see it sort of stands out. It's not similar to C and A, not similar to D and B. It's sort of a merger of the two, and it's here in the middle. But if we used Pearson instead, we would really see a very different grouping of our samples. The row clustering is not changed, but the sample relationships are. we can see we have a night with 2 days and a day with 2 nights grouping together. And that's because Pearson will cluster your data, but Spearman will first take a rank ordering of that data and operate on the ranks to figure out the correlation. So it's going to be less, Spearman's going to be less sensitive to magnitude. So it's less influenced by this dramatic change here. And so that's really important because we don't want some really highly expressed genes to skew or influence the clustering of our samples. So that's why we use Spearman correlation for samples, but Pearson correlation for our genes. So what do the colors in our heat map really represent? I told you we have this scale equals rho, and I demonstrated for you that really gave us a dramatic visual heat map if we go back, right, where we can clearly see our clusters. But why is that? Well, it turns out when we set scale equals to rho, it actually takes the underlying data that was passed to heatmap.2 and it calculates a rho z-score, all right? So these are standard deviations from the mean, basically. And so in that case, we can see very clearly these patterns for each gene across our samples. Compare that with if we set scale equal to none. This may not have been as apparent when we did it with all of our data, but hopefully with this toy data set, it's more clear that now, even though it's the same data being passed to heatmap.2, the visual implications are really dramatically different. Now we're plotting our actual data. We did not scale it. Scale equals none. And so this is the log2 expression. And what I can see is even though it's the same initial data set that was operated on, we have a very different look here. Now our eyes are drawn to gene C and gene D, C and D, because they're the most highly expressed. All right. We also see a little bit of gene E here because of course it comes up really high later, right? So that is to say that the choosing your scale here affects whether you display your actual data or a rho z-scores of your data. And if you do scaling by the rho, that really puts the emphasis on comparing between samples and understanding how genes change across your samples, irrespective of the magnitude of their expression. Whereas if you do scale equals none, it really puts an emphasis on comparing between genes. In other words, kind of comparing vertically, right? And so what tends to stand out when you don't do scaling is you tend to see the most highly expressed genes in your data set, which in this case are C and D. And it becomes more difficult to see nuanced patterns like we see with B here that actually does change in a circadian rhythm. You can see it here, it's light blue and dark blue. That's much more apparent if I scale by rho. This is gene B here. Okay, so hopefully you understand why we choose these scaling options, how they influence what we see, and why we do certain correlations when we're operating on rows or columns. Okay, I prefer to scale by rho because I'm usually trying to highlight patterns of genes that are behaving in a similar way, irrespective of the magnitude. I don't care if they went from 1 to 3 or 5 to 9. I just care that they went up. All right, so now back to our script. We've made the heat map, and I want to just kind of emphasize that you should play around with this heat map function and check out some of the options yourself. I am going to move to the next section, which is to make interactive heat maps. And I'll just emphasize that these use packages like Plotly or D3 to create heat maps that look like this, but when you mouse over, you get a tooltip pop-up that tells you about the genes, and you can select and zoom in on different parts of the heat map, either certain rows or certain columns. Pretty cool. Unfortunately, it seems to be broken for me, and I think that's because I very recently updated R. So if I try to run heat maply, I just kind of get stuck with this stop sign and it doesn't actually plot anything. And if I try to run D3 heat map, I'll stop this now, it will plot something, but not correctly. And so I suspect whatever this issue is will be resolved soon with an update to one or more of these packages. But I'll leave the code in here because eventually it will work. And if it doesn't work for you right now, it may be something you want to come back and try later. So the D3 heat map makes a heat map that I can't see, gives me the clustering results, but if I select, it then all of a sudden displays and it zooms in, and now I can see colors, right? So I don't know what's going on with these. They'll open in your viewer, not in the plots tab. Okay, moving on. I have a couple optional code chunks here. One is how to simplify your heat map here, and the other is how to pull out modules of co-regulated genes and export those modules for analysis downstream. So I'm going to move through these kind of quickly because they are relatively easy to understand, and they're things you can try on your own, and some of them are optional. Simplifying the heat map, what do I mean by that? I just mean that there are times when you may want to group all the samples together for one of your groups, like a cage of mice, together to represent them as a single column. And if you want to do that, this is particularly useful if you have a lot of different conditions or treatments. The heat maps can get huge if you show every replicate. So you can group your replicates together, and you can do that using the average arrays function from Lima. And what essentially is happening is it will group together the data for any column that has the same name. So the first thing we have to do is give our columns the same name based on their group assignment. Easiest way to do that is to take advantage of our targets dollar group, which if you recall is just healthy, 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 disease, 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 disease. So we can now assign those to the column names of our heat map and then use the average arrays function to operate on that and produce a new averaged data matrix that can now be put into our heat map function up here, the static heat map one, to produce that result. And I'm going to just get rid of the clustering of columns because, of course, now there's only 2 columns. And, of course, that's not so interesting because I only have 2 groups. But it may end up being interesting or useful for you down the road. All right. So that's the other code chunk beyond making the interactive heat map is making this simplified heat map. Okay. All right, now we want to actually be able to cut modules out of our data set. And to do that, I'm going to remake the heat map that we had before. And this is going to tie in very closely with what we have to do in the next lecture, which is functional enrichment analysis. So I'd like to be able to slice out this pink module and this orange one to be able to work with them downstream, right? I'm going to assume, and it's a fundamental assumption in biology, like I mentioned earlier, that the genes that are going up with infection, or down in this case, sorry, are fundamentally sort of functionally different or distinct from those that are going up. So what I'd like to do now is go ahead and pull out these modules of co-expressed genes. So to do that, I'm going to first capture the names of my module assignment. There they are. That's the genes, of course. And put them into the names of my module color. So it's going to make a new named vector here that looks like this, where each gene now has its color. You can see where this is heading. This will allow me to cut out any genes that fit into one of these colors, this module or that one. I'm then going to convert this to a list and turn it into a tibble. All right. Take a look at this tibble. And it is just a really, really long data frame where each column is a gene symbol and each row is its assignment to one module or another. All right, so this is a perfect case for pivot because that really is just 2 variables, gene symbol and module assignment spread out across a whole bunch of columns. So I'm going to pivot that entire thing, all 2,314 columns, and assign the names to gene ID and the values to a column I'll call module. Let's take a look at our new tibble now that's been pivoted, and we can see that now we have something that looks better, gene ID module assignment. Now that I have that, I'm going to go ahead and actually mutate it to add my colors as another column. And there's a unique way I can do that by using the case when function, which will let me take my data, mutate it, and create something called module color, a new column. And that module color, the values in that column will be this color if the module column equals 1, or this color if the module column, remember the module column is this one here, if the module column equals, the value in the module column equals 2. All right, so I'll run this and I'll take a look at what I produced. And now I see I've got this nice data frame, tidy data frame with gene ID, module, and the color assignment. So this will be really easy for plotting now. I can even see how many genes fall into each module if I want to. Here they are. It's roughly split, but there's more genes in that second module. And now I can choose exactly which module I want just by choosing that integer, and then carry out a subsetting operation, albeit a somewhat complex one, and we'll just kind of run it and you can visually parse it on your own. And once I have that module, I could redo the clustering of the rows if I choose, and redo the heat map. Okay, so we're good. It's produced. I'm back to my red-blue coloring because I choose module colors 2 instead of 3. And I can see, indeed, I pulled out the genes that go up with expression. Perfect. If I had changed this to module 1 and I reran that subsetting operation and the clustering of the module itself and then the heat map, I would now get a new heat map of the genes that went down in infection. Perfect. The last thing I have to do in this part of the video is just actually get the genes and their data printed out to a file on my computer, because a lot of times you're going to want to have just these genes that went down, or the previous heat map, just the genes that went up with infection. You're going to have those pulled out in separate spreadsheets really to be able to look through them yourself and also carry out some downstream functional enrichment analysis that again we'll do in the next class. So I'm going to use, again, I'm going to take this, use this Tibble function to make a data frame out of my module symbols, and it involves, again, another subsetting operation that I won't just sort of verbally walk through each step. I'll have you look at that on your own. And then I use that set of module symbols to subset my total data set of differentially expressed genes, the 2,314. I then convert that into a Tibble with the row names set to a column called gene symbol. And now I have a data frame here that looks great. It's got about 1,000 or so rows. These are all the genes that went down in disease. And I'm ready now to move forward and print that out to a spreadsheet. Okay, and I could just do that using the right TSV. All right, the final thing is just this idea of you don't need to go through all these steps. If you just have a set of 5 or 10 genes that you'd like to read in from your spreadsheet or text file on your computer, and just clustering or no clustering, turn it into a matrix, carry out clustering if you'd like, and make a heat map, right? So you could do this, what we call a priori, prior knowledge, picking out genes that you want to pick out. All right, we're going to stop there with this bit of the video, and we'll pick up at the end with an alternative for how we can cluster.